noticed that you look a little like your parents? Maybe your eyes look like your mother, and maybe your height look like your father, but still, you don't look like an exact replica of either of them. How does this happen? And how are characters transferred from the parents to the children? Or for that matter, why are there so many variations that take place within the members of a same family? So not to worry, because in today's session, we will get answers to all of these questions that is based on the chapter Heredity. Hi everyone, I am Kirti Ma'am, your biology educator, and in this session, we will break down heredity step by step in a very simple manner so that you are left with no confusion and no mugging up that you have to do. And not to worry about the notes, because we will be providing you with the class notes as well as DPPs absolutely free of cost. To get them, you simply have to click on the link in the description box given below so that you can be taken to Physics Wallah's app, and there you will get all the study materials absolutely free of cost in one place. Now you must be living under a rock if you don't know what Physics Wallah is. It is India's most trusted and leading learning platform where every year many students enroll in their courses and achieve their dream percentages. So let's get started. Now before we start, I would like to share a good news with all of you that now we have our very own WhatsApp channel by the name PW Foundation English. You simply have to scan the QR code to join in, where you will be getting obviously the notes and the DPPs, but apart from that there will be PYQs, sample paper solutions, mind maps and all the important updates that you need to know as a class 10th board aspirant. Now talking about the topics that we will be covering today, firstly we will have an introduction to the chapter like we do always, followed by inherited and acquired traits. Then we will understand some important terms that you need to know before we start with heredity. Then monohybrid and dihybrid cross, and finally we will conclude by studying sex determination in humans. Now talking about the name of the chapter, students, that is heredity, we should first understand the branch of biology that it comes under, that is called as genetics. So genetics is the major branch of biology that constitutes heredity as well as variation. Now you must have seen that people might say you that you look like your father, you look like your mother, or when a newborn is born, it is told that his nose looks like his father, eyes look like the mother. That means that somewhere offsprings or the children have resemblance to the parents, which also means that parents are somehow transferring some of the characteristics to their children. As you can see in the image, over here is a boy who has got some of the characteristics from the mother as well as from the father. The mother has curly hair, so the boy obviously also got curly hair. The father has green eyes, so the boy also got green eyes. That means something has been passed on from one generation to the next generation. This is the one generation to the next generation, something has been passed on. That is called as heredity. But do you also see when the characters are being transferred, they are not exact replica of the parents. That means the child is not an exact photocopy. You are easily able to distinguish that child from the parents, which means that along with something that is being passed on, there are some differences, and those differences, students, they are called as variations. So whenever heredity is taking place, alongside variations also occur, which is very necessary for survival, because we have already studied that in the previous chapter, that is how do organisms reproduce. Now talking about variation, you can also see it in a day-to-day life basis. For example, this is a bougainvillea flower and you can see it in multiple colors like yellow, red, purple, orange, white and so on and so forth. That means there are so many differences in the colors even though the plant is the same. Likewise, in case of earlobes, some people might have their earlobes free from the skin. Okay, this is the earlobe that is free from the skin. Some people might have the earlobe attached to the skin. Like my earlobe is actually attached to the skin. You might have free or attached earlobes. Some people have straight thumb, some might have curved thumbs, like mine is straight. Some people might have a taller height, some could have a shorter height. So there are so many differences in the characters that is studied under variation. For the time being, we mostly have to focus on how heredity takes place, that is how characters are being transferred from one generation to the next generation. Got my point? This is the basic definition. Now genetics is basically the branch of biology that deals with heredity, where the transmission of characters or features. What are characters or features? Basically something like your eye color, eye shape, body height, body pattern, nose shape, etc. They are all your features. They are transferred from one generation to the next generation. Now talking about variation, it says the differences in the characters or the feature in individual of the same species. For example, some person could have a taller height, some could have a shorter height. Some person could have green eye color, some could have black or brown eye color. Now talking about the next topic, that is inherited and acquired traits. Now you might think, ma'am, what are traits? We understood characters and features right now. You can for the time being understand that traits are also the same thing. Although there are some minor differences between characters and traits, but we shall not go into that depth so that you don't get confused. For the time being, remember, inherited and acquired traits somewhere means characters only, okay? Something that is called inherited traits. When you understand inherited, you will automatically understand what are inherited traits. See, you might have heard that your grandparents say that we have, protected this thing to be passed on to our next generation. For example, a piece of land, I will give it to my coming generation. So your parents got it. Now your parents think that I will give it to my next generation, that is you. Then you will also preserve it and pass it on to your next generation. So something is being transferred from one generation to the next generation. That is called as inheritance. Did you buy that land? No. Your ancestors got it. So that has been passing on from one generation to the next generation. That is called as inheritance. So inherited traits are also those traits that are passed on from one generation to the next generation. For example, obviously the attachment of earlobes, different types of nose shapes, eye color, height, body pattern, body structure, all of these are inherited traits because they are received from our parents. And simply as we received it, we will also pass it to our next generation. Do we have a say in it? When you are in the mother's womb, do you say that, mom, I want my body height to be this much, I want my eyes to be almond shape, I want my hair to be of this particular pattern? No. Because they are received from the parents, and as and how you pass it to the next generation also. They are called as inherited traits. You have no contribution in them. But on the other hand are acquired traits. Acquired means something that you have earned. For example, if you study well, you will earn good percentage in class 10th. That means you have acquired that because of your hard work or efforts. So acquired traits are basically all those characters or traits that you have acquired in your entire lifetime. You were not blessed with these traits since birth. For example, swimming, dancing, driving, a tattoo on your face. All of these are what you have earned during your lifetime. You were never born a great swimmer or a dancer or a driver, or you never were born with so many tattoos and piercings, but you got them. Will you be also able to transfer it to the next generation? No. You might be a good dancer, but your parents might not be as good dancers. And likewise, your children could also be not such great dancers. Which means this is what you have learned and earned in your lifetime. They are not received from the parents, and they will also not be transferred to the next generation. This is exactly what is written. Please make a note of it because they are several times asked in the exams in the form of differences, and they are basically like a low hanging fruit. If you learn them well, you can score good marks also. Talking about inherited traits, something that has been received from our parents. For example, they are because of sexual reproduction, can be inherited because they are received from the parents and transferred to the next generation. Example, skin color, eye shape, height, etc. Talking about acquired traits, these are acquired after birth during the lifetime of an individual. They are neither inherited, you never get these traits from your parents, and they cannot be transferred to the next generation. For example, swimming, dancing, piercing, tattoos, etc. are all examples of acquired traits. Now, before we jump on to heredity, the principles of heredity, Mendel's laws, Mendel's genetics, we should first understand some terms so that all of our journey coming up is very smooth. Okay, so we will first cover some basic terms like chromosomes, DNA, gene, and chromatin versus chromosome. Although we have covered this in the previous lecture also, but if in case you have missed out on that, I will just give you a quick recap over here so that you are well versed with these terms. You know that every single life form has the basic unit that is called as cell. So every single life form will have a structure called as cell, and inside the cell there will be a nucleus. So if I have to take out this particular structure, that will be called as nucleus. So I have just taken the nucleus out over here, okay, which is also the brain or the control center of the cell. Now once I open this nucleus up, I will find a structure which looks like this. This structure is actually called as chromosome. This structure is actually called as chromosome, and this chromosome is made up of two constituents, that is called as DNA, as well as protein, that is called as DNA as well as protein. So this chromosome is actually made up of DNA plus protein. Got my point? If I have to take the DNA out, okay, if I have to take the DNA out, I have just drawn a structure over here which looks like double stranded, which has two strands over here. As you can see, this is one strand and it is crossing the other strand over here that you see. So these are DNA are basically double stranded structure. There are two strands in DNA. Also, you should know the full form of DNA, that is called as deoxyribonucleic acid. Okay, this is your DNA. Got my point? This is almost like a thread only, but two threads which are crisscrossing with each other. Also talking about proteins, beta, these proteins are somewhere present on the DNA that helps in tying up the DNA or coiling up the DNA, because DNA is a long thread. So it has to be coiled up very tightly so that it can be put inside the nucleus. So we also have proteins around it. But for the time being, we should not focus on proteins because they are not relevant at this particular stage. We are more concerned about the DNA. But this DNA actually has all over genes present on it. There are obviously some genes present on it. For example, I say there is this particular segment, that is a gene. Over here is another segment, that is also a gene. Then we have some gene over here also. So your entire DNA is packed with multiple genes. Now ma'am, what are these genes supposed to do? These genes are not the genes that you wear. These are the genes that control every single thing, whether it is physical thing or internal thing going on in your body. Everything is controlled by genes. Okay. For example, this gene could be of your hair color. Some gene could be of your eye color. Some gene could be of your height also. So these are the different genes, but genes control each and everything, whether it is physical or internal. So now you got the point. Cell has nucleus, nucleus has chromosome, chromosome has DNA, and DNA has multiple genes, and each gene will control a specific character. So every physical attribute that you see is contributed by the gene itself. Now talking about chromosome. Right? Chromosome, when we talk about, there are certain terms that students get confused in. That is chromatin versus chromosome. This is very frequently repeated. But to know it, we should first understand this with the help of an example. Ma'am, what is chromosome? What is chromatin? Chromosome, you just now studied a structure that somewhat looks like this only. And it also has DNA and protein. Yes, you are right. But to make it easier for you, we should first understand with the help of a example. You must have seen Maggi, because all of us are very fond of junk food. So I think every student must be aware that Maggi noodles look somewhat like this, which are uncooked. They're very, very tightly packed, beta, very stiff, very tightly packed. Got my point? And once these are cooked, they look very, very elongated, very, very stretched apart, and each noodle is very beautifully seen. Got my point? So exactly like that, beta, whenever we see a tightly packed structure of DNA, that tightly packed structure of DNA is called as chromosome. And once that structure, beta, it is loosely present, that is called as chromatin. Understand this now. See, this is over here a loosely thread present of DNA. So this is your DNA that is present, and somewhere on this DNA are again some loosely present proteins. So these proteins are not doing anything. They are just loosely present, and DNA is also very, fully extended. This is called as DNA, which is fully extended. It is not coiled at all. This is DNA. Okay. Now, once beta this DNA decides to coil up, once this DNA decides to coil up, what will it look like? It will somewhat look like this very tightly packed structure, like a telephone wire. This is how it looks like. And once it is coiled, it will ultimately define to look somewhat like this. A structure that you just saw, it will look somewhat like this. Okay. So once they just coil up, they make structures which are called as chromosomes. So it is nothing else only, it is simply your loosely arranged DNA which just coils up to form chromosomes over here. So these structures that you get, they are your chromosomes over here. But ma'am, when will the chromatin be present? When will the chromosome be present? Let me tell you, whenever you see that a cell is in relaxed state, whenever a cell is in relaxed state, it is not doing anything, it will be present in the form of chromatin. It will be present in the form of chromatin. So when the cell is not dividing, chromatin is present when the cell is not dividing. Okay. Not dividing cell, and when the cell starts to divide, when it becomes or enters in the cell cycle, in the cell division, it will ultimately form chromosome. Got my point? So in both the structures beta, over here also DNA and proteins are present, and over here also DNA and proteins are present, and in none of these cases the properties are changed. Did the DNA change over here? No. Did the proteins change? No. Did the DNA change over here? Protein changed over here? No. Everything just remained the same, just the arrangement differed. That is called as the difference between chromatin and chromosome. When it is fully extended, it is called as chromatin. When it is packed, it is called as chromosome. Now, we should first read these points because please make a note of it that they can be asked several times in your exam. A chromosome is a thread-like structure present in the nucleus that becomes clearly visible during cell division. Very important. So when do you find chromosome beta? During cell division. Chromatin is the loose and uncoiled form, that is very extended. It is present when the cell is not dividing, or you can say that the cell is actually in relaxed state. DNA is the genetic material. Why it is the genetic material? Because it carries hereditary information. And what is that hereditary information in the form of? It is in the form of genes. So gene is a small segment of DNA. A gene is a small segment of DNA that will carry all the information for a particular character or a trait. Please make a note of it, like we just now discussed, that a particular segment of DNA beta might have multiple genes. This was also a question that was asked some years back, that does a DNA have only a single gene or it has multiple genes? It has multiple genes. Coming to our next term, that is called as haploid or diploid, and a lot of students get confused in this. Let us try to understand this one by one beta. Firstly, let us understand the term that is called as haploid, and then we will go for the term diploid. Okay, one by one we will understand both of these things with the help of a diagram. So in the first case, let us assume that there is one particular chromosome over here. Then we see that there is another particular chromosome over here, and then we have another chromosome over here. Let us also label them. This is chromosome number 1, this is chromosome number 2, and this is chromosome number 3. So over here beta is a cell which has all these different chromosomes. The blue chromosome, that is chromosome number 1, it is only present in one single copy. It is only present in one single copy. Chromosome number 2 also in only one copy. Chromosome number 3 also has a single copy. So a particular cell which has single copy of each chromosome is called as a haploid cell. Got my point? It is called as a haploid cell where every chromosome is present in a single copy. But now assume a cell which has two copies of each chromosome. For example, chromosome number 1 has two copies. These are the two copies, first copy, second copy. Likewise, chromosome number 2 also has two copies, first copy, second copy. Chromosome number 3 also has two copies. Chromosome number 3 also has two copies, first copy, second copy. So in a cell where there are two copies of each chromosome, like you see, first copy, second copy of chromosome number 1, first copy, second copy of chromosome 2, first copy, second copy of chromosome number 3. So every chromosome has two copies. It is called as a diploid cell. In fact, in the term diploid, the di word actually means two. That means every chromosome will have a duplicate of its own. Every chromosome will have a replica of its own. That is called as a diploid cell. Also, in this particular case beta, in case of haploid, the chromosome number is represented by N. Chromosome number is represented by N. Okay, now think of it. Over here chromosome number is by N. So let us see what is the N value over here. How many chromosomes do you see over here? Chromosome 1, 2, 3. So simply we can say N is equals to 3 in this case. It is very, very simple. Okay, N is equals to 3 in this case. In case of diploid cell, the chromosome number is represented by 2N. It is represented by 2N. Now think first beta, what is N? What is N over here? What is the N value? Firstly see, it is beta 3 because chromosome 1, 2, 3. So we have 3. but we have to represent by 2n. So what we are going to write 2 into 3, that is overall 6 chromosomes are being present over here in case of a diploid cell. That is how we have to mention. So haploid, diploid, I hope all of these terms are clear to every single one of you. Let us read the definition also. A cell having one set of chromosome, that means it is simply mentioned a cell which has only a single copy of every chromosome is called as a haploid cell, which is represented by N. Talking about diploid cell, a cell having two sets or two copies of each chromosome is called as a diploid cell, represented by 2n. That is about haploid and diploid. Now, one more important fact you need to remember is that all the cells in the human body are diploid except sperm and egg. All the cells in the human body are actually diploid in nature except for sperm and egg. And what are sperm and egg? As you know that we discussed last time that sperm and egg are actually the gametes. They are actually the gametes, the male and the female gamete, hai na? Male gamete is the sperm and the female gamete is the egg. But ma'am, what is so special about them that every cell is supposed to be diploid, but these two cells specially needs to be haploid in nature? That means they should have only one copy of each chromosome. Let us understand again this with the help of a beautiful diagram. See beta, firstly let us mark that this is a female gamete or the egg cell. This is the female gamete or the egg cell. So I'm gonna write female gamete, or we can say egg cell. Okay, and over here we have male gamete, or the sperm. Got my point? So these are the two gametes that we have which are supposed to be haploid. So let us see if they are actually haploid or not. Yes, definitely, because over here you see that there is one copy of chromosome 1, then you have other copy of chromosome 2, then you have also one copy of chromosome 3. Sperm also has one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3. So both of these cells have single copy of each chromosome, so they are definitely haploid in nature, as we can see. Now you know, once the gametes, that is egg and sperm, are produced, they will fertilize, they will fuse, and once they fuse, a single cell that is called as zygote will be formed. A single cell that is called as zygote will be formed. That is what we studied again in the previous chapter. So what will happen when fusion will take place, whenever fusion will take place, the female gamete will also contribute the chromosome. So see, the first chromosome from female gamete came over here. Likewise, the first chromosome from the male gamete will also come over here, and now you have two copies of chromosome number 1. Got my point? Likewise, chromosome number 2 will also come from the female gamete, right? As you can see, it has come, and chromosome 2 from the male gamete also comes and fuses. Likewise, chromosome 3 comes from the female gamete, as you can see, and chromosome 3 will also come from the male gamete. And now you see this particular zygote it has two copies of each chromosome. Reason being, one copy is received from the mother, one copy is received from the mother or the egg, the other is received from the father. One from the mother, other from the father, one from the mother, other from the father. So that is how the zygote has now two copies of each and every chromosome, because half the chromosome is contributed by the mother and half by the father, or I can say half by the female gamete and half by the male gamete. That is why the gametes are actually haploid, because when they fuse, they will automatically become diploid. The zygote will automatically become diploid in nature, and now it has two copies of each chromosome. That is why, to maintain the chromosome number, to maintain the chromosome consistency from one generation to the next, the gametes are supposed to be haploid in nature. Now one more thing over here, you need to remember okay, firstly we shall move to the next topic, then we will discuss. You see, in this particular zygote, there are two copies of each chromosome. Hai na? For chromosome 1 also you have two copies, for chromosome 2 also two copies, chromosome 3 also two copies are present. Now let us just take this zygote to the next slide. Okay, so here we have this particular zygote. And now over here you also see chromosome 1 2 copies, chromosome 2 2 copies, and so on and so forth. But for now we have just drawn two pairs. Okay. Do you see over here, one is from the mother, one from father, one from mother, one from father. But both of these chromosomes are actually, you know, since one is from mother, one is from father, both these chromosomes are actually the same in length, They have the same shape also, and they will also have the same kind of genes. For example, over here, if we say that there is a gene for eye color in the mother, exactly in front of it there will be a gene of eye color in chromosome number 1 of the father also. So this is the eye color gene, over here is also the eye color gene. Such chromosomes beta which have the same shape and which also have the same structure, size and the genetic arrangement or the arrangement of genes, they are called as the homologous chromosomes. Again I will repeat, both of these chromosomes where one chromosome is from the mother, one is from the father, and both of them are actually of the same length, same shape, same size, and both have a similar pattern of gene distribution. So if over here we have gene for eye color, over here the gene will be of eye color as well. Okay, it is not possible that the male has gene, so the mother has the gene of eye color over here and the father has the gene of eye color over here. That is not possible. Got my point? So it is definitely the gene arrangement is also exactly in the same manner. The height gene from the mother is over here and the height gene from the father is also supposed to be over here. Such chromosomes are called as, they are termed as homologous chromosomes. They are called as homologous chromosomes. Got my point, everybody? Let us also try to understand with the other chromosome pair that we have drawn. So over here you see that there is one particular chromosome from the mother, the other chromosome is from the father, both have the same structure, shape, size. So the gene distribution is also same and thus they are called as homologous chromosomes. So over here if there is a gene of particularly, let us say, body shape, so over here is the gene is of the body shape as well. And these homologous chromosomes are basically one contributed by the mother's side, one from the father's side, you can say one from the egg, one from the sperm. Okay, a pair of chromosomes that are similar in size, shape, structure, size, shape and structure, and also the same genes at the same place. Okay, same gene at the same place, they are from each parent, are termed as homologous chromosomes. Very, very easy to understand. But now also see, if in homologous chromosome, you know that over here both of them carry the gene for eye color. Both of them carry the gene for eye color. Let us say from the mother side the eye color is black. Let us say the eye color is black and over here the eye color is brown. Then what will happen? Will the child have one eye to be black? Will the child have one eye to be brown? Or is the situation possible or it is not possible? Now you have to understand the next term that is called as alleles. Okay, so again we have a homologous pair, one. Okay, one is from the mother side, one is from the father side. Got my point? Both will have the same arrangement of genes. Over here you have an eye color gene. Okay, this is the gene for eye, or let us say it is the gene for hair color. To make things easy, we have to think that this is a gene for hair color, okay? But this is the gene for hair color. So this gene that is present at the same location in the mother and the father chromosome, in the homologous chromosome, one could say that the hair color has to be black and the other could say that no, the hair color has to be brown. So over here you see, but a gene is one, but the variations are different. The gene is of hair color, but there are multiple variations. For example, if you go and buy iPhone 16 or 17, iPhone 16 or 17 model remains to be the same. Let us say 17 only this year it got launched. So 17 beta model is the same, but there are variations. Some have storage of 128, some have 256, some have 1 TB, 2 TB. So over here the model is the same, just that the storage is being varied. So you see the pattern is over here also, the gene is same, but the variation is seen in the kind of variation is in the color, right? So which means that there are different forms that can exist. These different forms of a gene beta, they are called as alleles. The different forms or different versions that you see, they are called as alleles. So one version of the gene says the hair color has to be black. The other version of the gene says that the hair color has to be brown. So alleles beta, they are actually different forms of a gene. They are different forms, you can say, or varied forms of the same gene present on the same position on the homologous chromosome. They are called as alleles. They are called as alleles, not allele. You don't have to call allele, it is allele. So alleles are basically different form of the same gene. Got my point? These are different forms of the same gene. Another example, if you want to take to make things even clearer, think of this to be the gene for your height. This is the gene for your height. This particular variant says that the height has to be tall. This says the height has to be short. Now what will happen? Now what will happen? Who will win? Whether the person's height or the child's height will be tall or short. Let us see from the next slide, but before that, beta, a small note that you can make just to make things clear for you. Let us also write, as you know, beta, genes control a character. Now you will understand things even better. Genes control a character. Got my point? For example, over here, gene is controlling hair color. Over here, gene is controlling hair color. Okay. But alleles control, beta, alleles control traits. Alleles control traits. Got my point? For example, the hair color also has a trait of black hair color or brown hair color. I hope everything is clear to you now. है ना? Over here you see that a gene is actually controlling a character. So the character is hair color. Okay, this particular gene over here is controlling a character, that is over here your hair color. But the alleles individually, they are controlling basically the traits. They are controlling the variant, different forms. That is, one says it could be black, it could be brown. So this is how exactly it is mentioned that characters controlled by gene, traits, that is different form, is being controlled by alleles. But again, the question comes, ma'am, if there are so many alleles, black hair color, brown hair color, then how will it happen? How will the hair color of the child be? Will the child have half brown hair color and half black hair color? Or is it going to be, a mix of both the colors? Let us see how. There has to be some relationship that is being established between the alleles also. Okay, so beta, whenever there are two different kind of alleles, the one that expresses itself will be seen. Okay, for example, if you see, here is a boy, he has black hair color. His hair color over here is black. Now, if we go back and see his gene combination, over here is the gene for the hair color. Over here is the gene for hair color. One says that the hair color has to be black, and the other allele says that the hair color has to be brown. And surprisingly, over here you see that the child's hair color is actually black in nature. It is black in nature. So you saw that whenever two different alleles are being present, the one has expressed itself. Black has expressed itself. So the allele that expresses itself, that is stronger one over here, I can say. Amongst the two, the one that expressed itself is a stronger one. So the stronger one is called as the dominant allele. Okay. So over here, beta, what can I write? That since black colored allele express itself, it is called as the dominant allele. Dominant allele. Out of the two alleles, this was dominant because it was able to express itself. It was able to show its effect. This is called as a dominant allele. And the other allele that could not express itself, it was basically the ghar ka chota bachcha that was not able to express itself. So that is called as beta recessive allele. So the brown hair color ka allele was actually the recessive allele. Recessive, which means a very weak allele. It is not able to express when dominant is present. So this is your recessive allele over here. Got my point? Now one more thing to remember, beta, that to represent dominant allele or recessive allele, in every time genetics you cannot write when this is dominant, this is recessive. So to make things easier for you, there are certain abbreviations that we use. Whenever we have to represent a dominant allele, what do is we pick up the first letter of the trait and we just write it in capital. So this is basically capital B. When you write capital B over here, the examiner will understand that you are talking about a dominant allele. Got my point? Likewise, when you have to write down the abbreviation for recessive allele, you simply have to use a lowercase letter of this particular trait. So I will simply write small b over here. I will simply write small b. So when you write small b, the examiner will automatically understand this is a recessive allele for the hair color. Got my point? Let us take another example of this, okay, because students can get confused. Think of this to be a flower color. Okay, let us see a flower color. In a flower color, let us say that there are two colors, okay. This is a gene for a flower color which has two colors. One is red and the other is pink, okay. Now red is actually the dominant allele and pink is actually the recessive allele. So how are we supposed to beta represent them? When we talk about red, since it is dominant, we are going to represent it with capital R. Okay. Now when we have to represent recessive allele, students might think that ma'am we have to write small p because the trait is pink, so we have just use the same alphabet in lowercase. But that is wrong. You cannot do that. Because once you have chosen the alphabet for the dominant allele, simply that has to be written in recessive. So for this particular recessive allele, you are not going to write small p, you are actually going to write small r. Okay, so it has to be small r because red is dominant, so you have written capital R. When you have to write recessive, you have to write small r so that we can know that this is for the flower color which is pink in color. You don't have to write capital P. So only one alphabet you have to write in dominant alphabet letter, capital letter, and for recessive in lowercase or small letters. This is how it is defined for your dominant as well as recessive alleles. Now talking about the definitions, the dominant allele is the one that is able to express itself in presence, either in presence or in absence, whether it is presence or absence of recessive allele, it doesn't matter, represented with a capital letter. Okay, talking about recessive allele, it expresses only in the absence of dominant allele. Ma'am, what does this mean? Don't worry, I'll just explain. It is represented with a small letter. Okay. Now, ma'am, what does this mean? This particular statement, it expresses only in the absence of dominant allele. Think of it. You have an elder sibling and you are the younger one, obviously. So, the elder sibling, as long as the sibling is present in the home, he will have his dadagiri. वो जो बोलेंगे वही होगा, है ना? So, if they order you to get a glass of water, you have to get it. If they ask you to do a certain thing, you have to do it. But you will become the Raja of the house when the elder sibling leaves for his hostel or college. So now you decide what you have to do. It is likewise the case for recessive. So recessive will express only when the dominant allele is absent. When the dominant allele is actually absent, when the recessive will express. Not to worry, again, I have got some examples for you to make you easily grasp this particular concept. उससे पहले just understand these two terms, that is homozygous and heterozygous, and then we shall go back to our main terms. Beta, homo over here means same. Homo over here, beta, it means same, and hetero means different. Hetero means different. Okay. So when I talk about homozygous, it means same zygous, and when I say heterozygous, it means it's different zygotic condition. Okay. Understand this very, very carefully with the help of an example. But first, let's take three cases. Case 1, case 2, case 3. Forget these three terms for now. Okay. Simply let us make the allelic combination for hair color. So we have these two, we have this gene for hair color. This particular says that the hair color has to be black. So I'm writing capital B. This says that the hair color has to be black. And this also say beta that the hair color has to be black. Okay, so both the alleles are absolutely same. One allele says black hair color, the other also says beta black hair color. Okay. Over here we see that again we have this particular condition where there is a particular gene for your hair color. And now both the alleles say that the hair color has to be beta brown. So the hair color has to be small b and small b, that is brown. Okay, and both are now again exactly the same. Both are absolutely friends with each other. You also say brown, I also say brown. That's it. Now talking about the third condition says that okay, I also have the hair color gene, but one allele says that for me the hair color has to be black, and the hair color that the other allele says is supposed to be brown. Now let us see beta what will happen. Very interesting cases. Okay, in case 1, both the allele says that the hair color has to be black. This is also saying black, this is also saying black. I also want to eat Domino's pizza. My friend also wants to eat Domino's pizza. So it is sorted. No confusion. है ना? So over here beta, since both the alleles say that they want to have black as a hair color, so over here the hair color hair color of the person or the child will be black. Got my point? Also, this particular condition is called as homozygous. Firstly, understand why is it called homozygous? Homo means same. Homo means beta same. So this is also saying same black color, same black color. So it is called homozygous because same. Again, now they are same, but both of them are dominant. है ना? Black is also over here dominant. This is also dominant. So both are saying the same thing and both are dominant also. So we have to write that this condition is called as homozygous dominant. This is called as beta homozygous dominant. Got my point? It is going to give the hair color to be black and both are going to be homozygous dominant because both have dominant alleles and since they are same, so homozygous. Now coming to another condition. Over here you see one particular allele says that the hair color has to be brown. The other also says the hair color has to be brown. So again it is sorted. The hair color will simply be brown. I also want to eat Pizza Hut. My friend also wants to eat Pizza Hut. So done. Decision is that we are going to Pizza Hut. Likewise, so the hair color over here without any confusion will be beta brown. Because both the alleles are on the same page. Both want the hair color to be brown. So very simple. But now when we have to write, what are we going to write? Homozygous definitely because both are same. Both are saying that the hair color has to be brown. So we are going to use the term homozygous. But now look carefully, beta, both of them, even though they are same, but they are recessive alleles. They are the small letter wala allele. They are both the recessive or the small letter wala allele. So we are going to use the term homozygous to hai, but it is recessive in nature. It is recessive. Got my point? It is recessive in nature. That is why it is called as homozygous recessive. I hope now the particular statement over here is clear to you that recessive allele will express only in the absence of dominant allele. Recessive will express only in the absence of dominant allele. So over here beta dominant allele is actually absent. You do not see any dominant allele over here. क्या यहाँ पे dominant allele है? No. Both of them are recessive. So recessive will express only when both are recessive. That is what was the meaning of the previous definition. Now talking about the third case beta, look carefully. Over here you see that one allele says that the hair color has to be black. The other says the hair color has to be brown. Again, what will happen? Half brown, half black? Or a blend of both colors? No. The color that will be shown in the hair of the child beta will be where the allele is dominant. So the hair color over here is actually dominated by your black hair color because black is dominant. So hair color of the child will be black because black is dominant. As you can see, capital B, it is dominant, so hair color will be black. And recessive will not be able to express beta because recessive जब भी dominant के साथ होता है express नहीं कर पाता. Recessive will only express in case of homozygous recessive condition. Both have to be in recessive state so that it can express. Got my point? Now talking about this particular condition, it is called as heterozygous because both the alleles are different. Both the alleles want different properties. So this is going to be heterozygous condition. This is the three cases that you need to remember. If you understand this once, all of your genetics so far coming up in the slides, monohybrid cross, dihybrid cross, it will be so clear to you. So please, even if you are confused, beta, rewind karke, watch it again so that you can, improve these concepts. If you are confused in these terms, to aage bhi you will also get confused, okay? Now talking about some terminologies, that is genotype and phenotype. Don't worry, last terminology and then we are more or less done with the terms. But there are two terms, genotype and phenotype. Okay, let us see. Over here we have again three cases. Now you must see, ma'am, so many cases, every case you have drawn. Okay, so let us see first case. Beta, in this over here, let us see, there is a hair color. Okay, this says capital B, this says capital B, this says this says small b, and this also says small b. This says capital B, this says small b. Now what will happen, ma'am? Let us see. I think most of you will be able to answer. If the combination is of capital B type beta, if the combination of capital B type, that means the chromosome have this combination of genes. This is beta the combination of genes, this is the combination of the genetic makeup. So genotype is what the chromosome combination is. The genotype is what the genetic makeup is. How the combination of genes or alleles are present, that is called as the genotype. Okay, so this will actually give the hair color to be black. This will give the hair color to be black. Now this black is actually the phenotype, what is physically visible. P for phenotype, P for physical appearance, P for phenotype, P for physical appearance, what we are able to see from outside, and G for genotype and G for genetic makeup, what is present in the genes or the chromosome combination. Got my point? Now tell me one thing, beta, are you able to see the genotype? No, you are only able to see the phenotype of how a particular thing is looking physically. My hair color is black. That is my phenotype, that is what I can see. But can I see what is my genotype? Do I have capital B or capital B small b? I cannot tell, right? Likewise, whatever is your hair color, what you are able to see is the phenotype. The physical appearance is what you can see. You cannot see what is the genetic makeup of it. Everybody, I hope it is clear. Now look at the second combination. It is saying small b, small b. Now small b, since both are recessive, it is going to give the hair color to be beta brown. So the phenotype of this particular child will be brown. That is, physically it will have brown hair color. Okay, and the genotype will be small b, which obviously you cannot see. This is in the cell, because you know chromosomes are present in the cells only. So you cannot see a cell, let alone the nucleus and the chromosome. You cannot see genotype, but you can see phenotype. Talking about the third case, we have another combination, that is capital B and small b. Okay, let me rewrite it precisely, capital B, small b, and over here, since there is one dominant allele, there is one dominant allele of capital B, that means the hair color will be again black. It will be black because dominant will express and the recessive one will not be able to express. So this is how the phenotype is going to look like. So genotype is what is present in the genetic makeup on the chromosome, and phenotype is what is present physically, is what you can see actually. That is the thing about it, right? Physical appearance, phenotype, genetic makeup, genotype. Let us read all the terms one by one, because they could be asked again in your definition format. है ना? Homozygous dominant. Homo beta, it means same, and dominant mean that both the alleles will be dominant. For example, capital A, capital A. Capital A, again they are same, so homo, dominant means both are capital in nature. Organism having two identical dominant alleles. There are two alleles, obviously, and both are dominant. Homozygous recessive. Homo means same beta, recessive means both are, not able to express itself unless they are present together. So over here you see homo, again same, capital A, recessive written in small letters. Organism having two identical recessive allele, two exactly same allele, and both are recessive in nature. Heterozygous beta, hetero means different. So one allele is capital A, the other allele is small a. Organism having one dominant and one recessive allele, and in this case where one dominant, one recessive is present, the dominant one will express itself. Phenotype is the physical or the visible expression, whatever you can see physically, whatever is visible to you is called as phenotype. Genotype is the genetic makeup of allelic combination, you can say, of a particular gene, okay, and you cannot see the genotype obviously. Now let us try to solve some questions on the topics that we have covered so far. The first question is assertion reason type, and the assertion says a human child bears all the basic features of human beings, which is very true because our body structure is almost similar to our parents in the sense we have the same eyes, we have a nose, we have 2 ears, we have our limbs, which is similar to parents. So assertion is absolutely right. It looks exactly like its parents, showing little variations. That is not true because obviously there are variations that are seen. Even though the skeletal structure remains the same, but there is height difference, there is change in the eye color, hair pattern, hair color, and there are so many variations that can be easily seen, right? So the reason is wrong over here, so we have to choose assertion is true, but reason is false, and I can choose option C on the basis of this particular assertion reason. The next question that we have says the statement that correctly describes the characteristics of a gene. So you have to choose the right statement, the correct statement about gene. In an individual of a given species, a gene is located on a particular chromosome. That is absolutely true. You know that the gene which controls the basic characters reside on the chromosome. So, abhi ke liye option A seems correct. A gene is not the information source. It is saying a gene is not the information source, which is not true because a gene is the information source for making a protein. Each chromosome has only one gene. That is not true. I told you that a gene that is present on a chromosome is not the only one. There are so many that are present on a particular chromosome. All the inherited traits in humans are not controlled by gene. When we use the term inherited, it automatically means that there is something or the other related to genes. So definitely option D is also correct. So I can choose option A to be the correct option. The next question says that human population shows a great deal of variation in traits. That is absolutely true, because if we talk about humans all around us, we can see that every human has different patterns in their phenotype only, let alone genotype. So there are definitely many variations that can be seen in physical appearance also. All variation in a species have equal chances of surviving in the environment in which they live. That is not true, beta. All the variations do not guarantee good survival. There can be variations which are certainly harmful to the person, and there can be certain variations which can be beneficial to the person. For example, let us take the case of coronavirus. When the pandemic happened, there were certain people who had a higher immunity which were able to survive and fight the coronavirus. But on the other hand, there were certain humans who were not very able to fight the virus because their immunity was low. So the variation in the immunity was not always helpful. Only the higher immunity ones were able to survive, so it was beneficial. But what about the lower immunity variation that was not able to survive? So definitely it was not beneficial. That means not all variations are, helpful. So we can eliminate the reason, but obviously the assertion happens to be correct. So we have to choose assertion is correct, but reason is false. That is option C is the correct option. Now coming to Mendelian genetics. Now you might think that, ma'am, we have understood the term genetics, but what does Mendelian mean? But a Mendelian actually comes from the name of a scientist who is Gregor John Mendel, and Gregor John Mendel is actually the father of genetics. The father of genetics because he established the laws of genetics. He studied genetics when nobody knew about it. That is why when we study genetics, we also use the term Mendelian in front of it because it resembles that it was being done by Mendel. Now talking about beta Gregor John Mendel, as you can see in the picture, he was not a biologist. Very surprising, right? Because he gave the laws in biology, he gave the laws of genetics, yet he had no knowledge of biology. He was actually an Austrian monk. So when he was living in his monastery, there were certain plants that were growing around him in the monastery. And suddenly he caught his eye on the pea plant, the pea that you specially eat in winters. And that pea plant, he found that there were certain characters that were found on extreme ends. Like if the plant was tall, then it was only tall, and the other plant was very short heighted. There was no medium heighted plant. Likewise, if a plant had purple flower, it only had purple, or the other had white. There was no medium color. So he was very fascinated by how are these patterns being studied, how are these patterns being followed. Why is there no middle character that is seen in the plants? So he started studying on those pea plants without having any information about what is DNA, gene, chromosome. He just did multiple experiments on pea plant. So we can say that pea plant was sort of his guinea pig because he did many experiments on it. And the experiments that he conducted, they are called as hybridization experiments. Not to worry, we will understand this, what is the meaning of hybridization in the coming slide. But he did all of those experiments between 1856 and 1863, and later on his work also got published in 1866. But let me tell you students that, when his work was published, it was so, disregarded because people did not believe in science, people did not believe in scientific information. They did not even think that the new generation is actually being produced by sexual reproduction. So his work was almost remained unknown during that time. It was much later that his work got rediscovered by few other scientists, and then he got all of the recognition that he deserves. In fact, we are studying it, and not only studying it, his laws are mostly applicable in many cases of genetics, okay? So he explained how traits are passed on from parents to the offspring, and he also gave the laws of inheritance, which are very essential for you to understand because questions are 100% asked from this particular laws. Firstly, let us understand the term hybridization, because in the coming slides, even when you read your books, you will find the term hybridization or cross being repeated multiple times. But when we say hybridization or cross, it simply means sexual reproduction. It simply means sexual reproduction. That is, in a layman term, you can remember that sexual reproduction, when we study Mendelian genetics, it is also termed as hybridization or cross. Now, when we talk about hybridization, you will see a plant with particular genotype was crossed with the plant of other genotype. A plant which was dominant was crossed with the plant which was recessive. So to understand hybridization, I have taken two particular flowers. Okay. Now you very well know what happens in case of sexual reproduction is that the anther produces pollen grains. The anther actually has pollen grains, and those pollen grains have male gametes. So over here, this particular anther beta, it produced the male gamete which was present inside the pollen grain. So now this whole pollen grain will be actually carried, this whole pollen grain, by the action of wind, I can say, or by the action of air, water, it will be carried and then it will be taken to the stigma of the other particular flower. Once it has reached there, it will move through the pollen tube. The male gamete will ultimately reach the ovule present over here, and once it reaches the ovule, they will once it reaches the ovule, there will be egg. When they will fertilize, there will be formation of a zygote, and once the zygote is formed, beta, it will further form embryo. Okay, meanwhile, you know that the ovule will actually form the seed. So when the ovule forms the seed, beta, after sexual reproduction, sow it in the soil and then he will get a new plant. So all that he did was hybridization between pea plants for so many years. He produced so many new plants. All the things that he did was to simply take away the pollen grain from one particular flower of a pea plant, and then he used to manually put it on the stigma of the other particular pea plant, so that pollen tube will form, male gamete will reach, and it will fertilize with the egg, ultimately form a zygote and an embryo. Embryo will obviously form into a new plant, and then seed formation takes place. So once the seed has been formed, beta, this seed can be used to sow in the soil. That was the whole purpose of hybridization. So I hope now you understand, whenever you see the term hybridization or cross, you should be familiar that it is being considered as sexual reproduction. Now Mendel did not know that pea plants could be so beneficial for his experiments. He simply found them very fascinating, so he started conducting his experiments on them. But it was, you can say, fortunately a good thing for him, because pea plants as a model were very, very beneficial. There were certain reasons why it was so. Obviously, firstly, Mendel conducted his experiments on the pea plant, that is scientifically also known as Pisum sativum, that is called as Pisum sativum. Okay, you might be asked in one marker, what is the scientific name of the pea plant? Pea plants show many clear contrasting characters. I will just explain it to you in a bit, that what does contrasting characters mean, but for now you can simply assume that when we say contrasting, that means different or opposite. For example, if I say black color, there is white color, so they are very contrasting, right? So for pea plants, there were characters that were extremely contrasting in nature. For example, if I talk about plant height, so plant height could be tall or it could be short. So they are contrasting, they are opposite, they are different. So there were many contrasting characters that were seen. For example, again, if we talk about seed shape in the pea plant could be round or it could be wrinkled. So again, it is a shape, but there are two contrasting traits, and there were 7 different contrasting characters that he chose, which we will just study in the coming slide. Pea plants are actually bisexual in nature, so self-pollination is easy. What does that mean? Since they are bisexual in nature, simply over here the pollen grains can transfer from the anther to the stigma. So that is also a benefit, that you know, self-pollination can easily take place. Next, cross-pollination can be done. Obviously, Mendel did cross-pollination also by simply taking the pollens from one particular anther to the stigma of the other pea plant. Pea plant have a short life cycle, very important beta. If they have a short life cycle, that means within a very less time duration, many pea plants could be produced and they could be easily studied for their desirable characters. And they also produce a large number of seeds. So more the number of seeds, more the number of new plants that can be grown, and since they have very short life cycle, that means they grow up really fast. So we can study the characters easily. Pea plants are easy to grow and maintain. They do not require a lot of maintenance also. So once they are let alone for growing. Now to understand contrasting traits, like this point particularly said that plants show many clear contrasting traits. What were they exactly? So contrasting, like I said, it simply means that there are two different or opposite traits. So Mendel studied 7 pairs of contrasting traits. 7 pairs of contrasting traits. Now what does that mean, 7 pairs? When I say a pair, it means that there are 2 items that are being included. That is what makes it a pair. So Mendel actually studied beta 7 pairs. Let us see the 7 pairs over here with the help of a picture representation. Now there is a first pair, okay, which has again the different traits. Second pair, third pair, fourth pair, fifth pair, sixth pair and seventh pair. So overall there are 7 pairs of contrasting traits that he studied. There were 7 characters and each character had 2 different traits. For example, when we talk about flower color could be purple or it could be white. If it is purple, it is dominant. If it is white, it is recessive. Likewise, plant height, it could be tall, it could be short. Tall is dominant, short is recessive. Seed color could be yellow, could be green. Yellow is dominant, green is recessive. Seed shape could be round, could be wrinkled. If it is round, it is dominant, wrinkled, it is recessive. Pod color, it could be green or it could be yellow. You must have seen it when you also buy peas, or if peas are let alone for a long duration of time, they turn yellow itself. Green is dominant, yellow is recessive. Pod shape could be inflated or constricted. Inflated means it is very khila and constricted means very murjhaya hua sa. Again, inflated is dominant, constricted is recessive. Flower position could be axial or it could be terminal. Axial is dominant and terminal is recessive. Now, for example, if we want to represent that purple is a dominant trait, so we have to use capital P. I hope you remember. When talking about recessive, you don't have to write small w beta. You will not write small w, you will write small p. That will mean that it is recessive and it is white in nature. Likewise, for tall, you can write capital T, and for recessive, you can write small t. Likewise, for yellow, it is dominant in the seed color, so capital Y, and recessive, so you will not write small g, you have to write small y. These are some tiny bits that you have to remember. Okay, seed shape round, again dominant, capital R, wrinkled, small r. Again, pod color green, capital G, yellow, small g, which means it is recessive. Inflated, capital I, that means it is dominant, small I means constricted. Flower position axial is dominant, so again capital A and small a for terminal. That is how you represent all the different combination of traits. Now talking about the crosses that Mendel did. First is monohybrid cross. Now what does monohybrid mean? Let us break down the term itself. Mono means one. Mono means one, and hybrid, again you know that it means sexual reproduction that is being done, right? So over here Mendel did the sexual reproduction process only involving one particular character. So out of these 7 pairs of characters, Mendel chose only one pair of character, okay, for the time being. You could think of it to be this one. Okay, this is also one particular character. You could choose this one also. You could choose this one also. But Mendel only at one particular time did one character, so which was this. He went with this one, which is most famously studied, although he did with all of it, but the most frequently studied is with the plant height. So monohybrid cross may you only have to consider one character and forget about everything else. Got my point? So that is what was being done in monohybrid. Again, a note that is being given is for parental generation, Mendel chose pure homozygous plants. Now what does pure homozygous means? Sometimes it could be also written in books that it is pure line. What does pure line or pure homozygous mean? But a pure homozygous simply refers to the term that it is homozygous. It is homozygous, or pure line is one and the same thing. It is simply a homozygous condition that it is a pure line. Now homozygous could be of 2 different types, as we all know. Homozygous could be homozygous dominant, as we all know, and it could be homozygous recessive. Okay, now if it is homozygous dominant, it could be anything. So it has basically supposed to be 2 dominant allele. For example, if I am writing capital A, that means it is dominant. If I am writing small a, it is recessive. It is homozygous recessive. So for the first generation of parents, Mendel chose homozygous or pure lines. What does homozygous or pure line mean? Either it is absolutely dominant or it is absolutely recessive. So whenever you see this in a question, this will also help you in solving the numericals, that pure lines or homozygous means they are either homozygous dominant or homozygous recessive. That is the parental condition, and that will also make things easier for all of you. Now talking about the parents' first case, when the first generation of parents was taken, again monohybrid cross में only one particular character has to be studied over here. So for the first generation, Mendel chose pure tall parent, and the other one was pure dwarf parent. Okay, pure tall and pure dwarf. Now think about the genotype. This is what you know as the phenotype. It is pure tall and pure dwarf. But what will be the genotype of this one? Pure tall. That means it is homozygous dominant. We just now knew that pure means homozygous. So homozygous tall, capital T, capital T. Pure dwarf will mean homozygous recessive. That means small t, small t. That was the parental combination, beta. That is called as the parental combination. So parents ka genotype now you are very well versed with. Now when these parents will form gametes, what will happen, beta? When the parents will form gametes, both of them will separate. So one particular allele will go in one gamete and the other particular allele will go in the other gamete. How did this happen? How did this take place, beta? How did this take place? Just give me a second so that I can erase this particular, yes. So how did this happen? Why did the two different alleles separate? Whenever gamete formation takes place, as you know, gametes are haploid in nature. They have only one particular copy of each chromosome. So when they separate in the time of gamete formation, one of the gametes will carry this capital T, the other gamete will carry this capital T. Got my point? Likewise, then this particular parent will also form gametes. What will it do? They will be separated. The alleles will be separated. So how will it look like? One gamete will have small t and the other gamete will have again small t. So both of these are now separated. Now talking about how these gametes will form the next generation, there is a possibility that this capital T could fertilize with this small t. This capital T could fertilize with this small t, or these could also fertilize. So let us see what will happen. Let us see what will happen if this capital T fuses with this one. It will form capital T small t. Again, this could also fuse with this. So again, capital T small t. This could also fuse capital T small t. And when they will fuse, it will form capital T small t. Now this generation that you just now receive is called as filial 1 generation, or it could also be termed as F1 generation. Got my point? Filial 1 or F1 generation, one and the same thing. But over here, did you notice that all the plants, beta, all the plants that you see over here are tall in nature? Why are they tall in nature? Because they are heterozygous and they all contain one dominant allele. So when there is one dominant allele, all of them will show the dominant character only, dominant trait only. Got my point? So over here, beta, in F1 generation, the phenotype, if we talk about, all the plants will look tall. Now Mendel was really surprised because he did not expect that all plants could be tall, because the parents that he took were pure tall and pure dwarf. So he thought that once the children come, they might be either a medium heighted one, है ना, or they could either half be tall and half be dwarf. But he never expected that all of them will look tall. So he was very surprised. He found something like this over here that you can see in a very beautiful pictorial representation. For the first generation, he took a tall parent and a short parent. Now what was he expecting? That in F1, all the children should either be medium heighted or half should be tall and half should be dwarf. But he found all of them to be tall. So again, that could, strike his mind that what is exactly happening in the plants? Why is the shorter one not being expressed? The short height one, why is it not able to express? So Mendel, what he did was, he picked up two different plants from the F1 generation and asked them to self-cross. Okay, so he picked up two different plants from the F1 and he crossed them. Got my point? Let us see over here also. He took two different plants. Any two plants he could pick, this one, this one, this one, this one, this or this, any two he could pick, and he simply sexually hybridized them. Okay, what does that mean? That is also called as selfing. So when we talk about selfing, it simply means sexual reproduction between genetically similar plants. So all of these are genetically similar. So he picked up two of them and he simply reproduced them sexually. So in the next case, he simply picked up two plants of the F1 generation and asked them to sexually reproduce. So it will be simply written as F1 and F1. What does that mean, beta? Now let us represent in the next slide that over here he simply took two plants from the F1 generation. Over here he took two plants from the F1 generation. So what can I write? Two plants from F1 into F1, and now they were acting as parents. Okay, what were the genotypes of the F1? All of them were the same, one and only capital T small t and capital T small t. All of them were heterozygous. Now when they are supposed to form the gametes, what will they form? Again, there will be separation of the alleles. So one of the gamete will have capital T, the other will have small t, and likewise over here in this case, one will have capital T, the other will have small t. Now these are the gametes that he received. Again, there is a possibility that either this could fertilize or either this could fertilize. Anything could happen. But just to make things easier, now we will draw a Punnett square to represent what exactly happened. What is a Punnett square, ma'am? Beta, a Punnett square is a square that will represent the genotype and phenotype of a particular offspring. Okay, so we have first drawn a square, and now let us draw a Punnett square with certain boxes. So these are the boxes. Now ma'am, how did you decide that we have to make 4 boxes? Let me tell you, count the number of gametes that were being formed. How many gametes are being formed over here? 1, 2, 3, 4. Of both the parents, totally there are 4 gametes. 1, 2, 3, 4. So you simply have to make 4 boxes in a square. That is called as your Punnett square. So I'm going to write this, that this actually represents your Punnett square. Now in case of Punnett square, beta, what will happen? You simply have to write the gametes of one particular parent over here. Let us say that this is the female gamete. Okay, this one is the female gametes. So we are going to write capital T small t over here. And now let us think that this is the male gamete. So we are going to write the gametes of the male over here. That is how you represent in a Punnett square. Now let us try to fill them up. What will we find over here? Once these two fuse, they will form capital T and capital T. Once they fuse, they will form capital T and small t. When they fuse, they will form small t and capital T, right? But you have to not write this as small t and capital T. Since you have a dominant allele, you will always represent dominant allele first. So whether you are making them fertilize, you have to always mention the dominant allele first. Then you have to write small t and small t. Now all of us will together write genotype and phenotype over here. See, this will look tall because it has both the dominant allele. This will also look tall. This is also supposed to look tall, but this one will look dwarf because it has both the recessive allele. So Mendel was again very surprised that in F2 generation, the filial 2 generation, the dwarf parent could also express because the dwarf offspring is also seen. So that is what exactly happened in the FN generation. In the FN generation, all of the offsprings were tall. All of them were tall. But in case of F2 generation, there was some which were actually dwarf in nature. So which means that the dwarf parents also express itself, the recessive trait also express itself, but only in the F2 generation. Now, on the basis of which we can also write the phenotypic and the genotypic ratio. So, let us write it together. First, we will write the phenotypic of the F2 generation. So, let me rewrite it in a better way. Phenotypic ratio. These are only for F2. Okay. And then we also have to mention genotypic ratio of the F2 generation. Let us see beta what is the phenotypic and the genotypic ratio. Talking about the phenotypic ratio, there are two different phenotypes that you can see, tall and again dwarf. So we are going to write the phenotypic ratio for tall is to dwarf. Tall is to dwarf for F2. Let us count how many are tall and how many are dwarf. You see 1, 2 and 3. They are tall and 1 is dwarf. So what we have to write? Tall are actually 3 and dwarf is actually 1. So the phenotypic ratio happens to be 3:1 for a monohybrid cross in F2 generation. Okay. Talking about the genotypic ratio, how many genotypes are there in the F2 generation? 1 is capital T, which means homozygous dominant. 2 are heterozygous, that are capital T small t. 1 is homozygous recessive. So let us write again the 3 different genotypes that we can see in the F2 generation: capital T, capital T small t and small t. Let us count how many capital T? Capital T only one homozygous dominant. Heterozygous how many? 1 and 2. So again, 2 we have to write that they are heterozygous dominant. How many homozygous recessive? Only 1. So we have to write again 1. That is 1:2:1 will be your genotypic ratio for a monohybrid cross in the F2 generation. Please make a note of it because phenotypic and genotypic ratios are often asked for a monohybrid cross and they are very, very important for you to understand. Okay. By the way, just one more thing that I have to let you know is these could be also asked in the form of a percentage. So let us see how to answer them. If we talk about phenotypic ratio, you know that in the case of phenotypes, we have 3 tall is to 1 dwarf. This is the tall is to dwarf the ratio. How can it also be asked that what is the percentage of tall? So what do you have to do beta? What do you have to do? Overall there are 4 plants. And out of which 3 were tall. So into 100 you simply have to do. Now you can just cross them up and what you will find 75% is the percentage of the tall plants in the F1 generation. This is actually for your tall plants. Okay. Likewise, they could also ask you for dwarf plants and how many dwarf are there out of 4? Out of 4, only 1 dwarf is there, right? Because this is your overall 4 and only 1 dwarf is present. So what do we have to write? Again, 1/4 into your 100 for percentage, which gives us 25% are actually your dwarf ones in the monohybrid cross of F2 generation. That is how you represent. Got my point? So please make a note of it. This could be very, very helpful when you try to solve some numericals as well. The same thing could be done for your genotypic ratio as well. This could also be done for your genotypic ratio as well. So what you have to do is now you have to comment down what will be the percentages of all of these different genotypes. That is capital T, capital T, capital T, small t and small t, small t. You know that the ratio is 1:2:1. I will just write it over here so that things are easy for you now. And comment down the percentages. This is 1 to 1. Overall, again, you have 4, right? So if I have to calculate for capital T, I'll write 1/4 into 100. So this will give me 25% are actually your homozygous dominant. For example, if I want to do it for homozygous recessive, what I am going to do again, 1 is homozygous recessive out of 4 into 100. Again, I will get it to be 25%. And now this particular one you have to let me know in the comment section that how you will calculate the percentage of heterozygous genotype in the F2 generation. Although I have made things very, very easy for you, but still you can answer them. Now coming to the same thing but represented in a form of a picture, that once you have got the F1 generation, all of them are tall. He simply crossed F1 into F1, which is also called as selfing, because in case of selfing, the plants with similar genotypes are being made to reproduce sexually. Now over here you have 2 different tall plants that are taken from F1. So let us say they are these 2 plants and they are acting as parents. This is acting as parent 1, this is acting as parent 2, and both of them are actually received from the F1 generation. So over here you will find that out of 4 plants, how many are tall? 3 are actually tall and 1 is actually dwarf in nature. So this is how you actually get your F1 and F2 in monohybrid cross. The same thing can be represented because over here is a Punnett square along with the ratios. So I have drawn it so that you can just take a screenshot in case you don't want to make notes. That is a bad thing, but still I'm helping you out to make things easy. Phenotypic ratio 3:1. Genotypic ratio 1:2:1 for monohybrid cross. Please make a note of this one because, like I said, very important. Now coming to the laws that he gave on the basis of monohybrid cross. Firstly, you have to remember that the laws are actually supposed to be remembered the way it was given. But since you cannot remember word to word, please do not make your own language. Whatever is given, try to memorize that because I have made it as simple as possible. Laws are designed to convey a certain statement, a certain message, and if you mold it as per your own convenience, that will not fetch you marks. So when you talk about law of segregation, it was given on the basis of monohybrid cross. Right after that, I have mentioned the law. So which means once he got the monohybrid cross, he gave the law. That is the law of segregation. But a segregation, it simply means separate. Like for example, you must have heard waste segregation in your home. Dry waste and wet waste, you segregate them. That is how it means for segregation. So it says during the formation of gametes, the pair of alleles for a trait will separate and each gamete carry only one allele. Now ma'am, this seems very confusing. Not to worry. Let us take an example. Okay, let us say we have a particular genotype capital T, capital T. Let us say we have a genotype capital T small t, that is heterozygous, and let us say we have homozygous recessive. So they are simply saying, the statement simply says that whenever there will be formation of gametes, both the alleles will separate, beta, both the alleles will eventually separate. So both the alleles will separate and each allele will move into one gamete, So they separated and each allele has moved into one gamete. This particular has moved into one, this has moved into the other gamete. Likewise, when they will form the gamete, they will also separate. So capital T will be in one gamete and small t will be in other gamete. Likewise, when they will form gametes, small t will be in one gamete and the other small t will be in other gamete. So that means whenever gamete formation takes place, both of the alleles will separate and each of the gamete will have only one allele. Each gamete has only one allele, na? That is what it is meant to convey. It is not possible that when gamete formation takes place that both the allele come in one particular gamete and the other gamete remains gameteless, alleleless. That is not possible. So whenever gamete formation occurs, both the allele will separate and each gamete will have only one copy of the allele. That is what it was being simply conveyed over here. I hope it is clear to everyone. Coming to the next law, that is called as law of dominance. Law of dominance, beta, it is simply saying that whenever there is a heterozygous condition, or you can say that whenever there is two different allele of a particular character, whenever there are two different alleles of a particular character, the one that will express itself, it will be called as the dominant allele. Because obviously it is the stronger one, it expressed itself, and the one that is not able to express, that will be called as the recessive allele. I hope it is clear to everybody that whenever there is a heterozygous condition, whenever there is a condition of two different alleles, the one that will express itself, it will be called as the dominant, and the other whose effect is masked or it is not able to express will be called as the recessive allele. As simple as that. These are the two things that you need to remember. Now coming to questions. When a pure tall pea plant, what do you mean by that? Pure tall pea plant. This is what I was concerned about, that I had to explain it. What is a pure tall, beta? What is a pure tall? Pure tall is this, because it is homozygous tall, hai na? So please write pure means homozygous, although we have mentioned, but still whenever you see things, make a note of it. So pure tall means that it is a homozygous tall pea plant. It is crossed with a pure dwarf pea plant. What does a pure dwarf means now? It means that it is actually a homozygous recessive. So when both of them were crossed, what is the question saying? The percentage of tall plants in the F1 generation and in the F2 generation, what will they be? They are asking about tall. So let us try to make the cross, okay, of F1, F2, both of them, although we just studied, but let us see. The alleles will be this in case of F1 generation. So we get capital T, small t, and all will be tall. So which means 100% will be tall in case of F1 generation. For F2, you simply have to self-cross these, and in this you will find that how many are tall, beta? If you have to talk about the percentage, you will find that 75% of them are tall in case of F2 generation. So in your F2 generation, 75% are actually tall. So what do we have to choose? 100% in F1, 100% in F1 and 75% in F2. So we see 100 over here. But this is not right, this is not right. Over here we see 175, 100% in F1, 75% in F2. Not this one again, 100% is not seen in F2. So the correct option happens to be option C. Now, question number 5, it says a cross made between two pea plant produces 50% tall and 50% short pea plants. Very nice question, beta. Please mark it as important because it is reverse asking you the question. Let us try to solve this one also. It says when the cross were made between two pea plants, we don't know what were the genotypes of two pea plants, so we are just going to mention a dash over here. The progeny was 50% tall, and 50% short. 50% short. Okay, got my point. Okay, 50% tall and 50% short. Now for this case, we have to find out the parental pea plant combination. What was the parental combination of the genotypes? But if you see 50 are tall, 50% are short, that means that one of the parents definitely will be homozygous recessive. One of the parent will be homozygous recessive. So definitely I can choose that one parent will be homozygous recessive. Now the other parent has to be tall so that the tallness could be come. So because of which I have to think if the other parent is tall beta, tall could be because of two reasons, homozygous dominant tall or it could be heterozygous tall. So now we have to confirm this out of the two parents which one will exactly fall over here, because about one of them we are sure. About one of them we are actually sure that this is supposed to be a homozygous recessive one. But what about this one? It could be either case. It could either be a homozygous dominant tall, it could be a heterozygous tall. So what will be the exact one? Let us try to solve this by answering, by putting both of them. Let's put both of them over here and then see what is the exact answer. See, we have for tallness, it could be capital T, it could be capital T, small t heterozygous. First, let's go for this one. So, I'll make a cross with this one. Okay. What will you find, beta? All of them will be tall. All of them will be tall. So, definitely you cannot go for this one. This one is eliminated. So, we cannot go for this one. Now, the option left is for the other one, that is heterozygous. For the heterozygous, again, let us make the alleles. Capital T over here, small t. And over here, we will have small t and small t. Now let us try what is happening. Capital T, small t, capital T, small t, small t, small t, and again we have small t, small t. So now I can easily see that there are 50% plants, which are actually tall because they have one dominant allele, and then 50% are actually dwarf because they are homozygous recessive. So to obtain the progeny where 50% is tall, 50% is dwarf, the parents need to be one heterozygous and the other needs to be homozygous recessive. So the correct combination of the answer will be capital T small t and small t, which is option C. So option C is the correct answer over here. I hope everyone is absolutely clear with this. Please try to practice questions like this because it is not necessary that they give you the parental genotypes and ask for the offspring phenotype and genotype. It could also be possible, like in this question, they gave you the offspring phenotype and now they are reverse in a manner asking you the genotype of the parents. These are very frequent questions that can be asked and they could be confusing if you don't practice them well. Next question says, in human gene B is responsible for black eyes. Okay, gene B is responsible for black eyes. Got my point? Okay, and gene B, I mean this particular allele is responsible for brown eyes. Okay, so if it is saying but gene capital B is responsible for black, this is for black, and it has also mentioned in the bracket that it is capital, so which means it is dominant also. Black is dominant also. Very good. We have got this point. Then it is saying that the gene small b is responsible for brown, and since it has mentioned in the bracket that it is small b, which is also signifying that it is recessive in nature. So it is recessive. Then the color of eyes of the progeny having the gene combination small b or capital B small b will be black only. Let us see beta. If it is small b, that means it is homozygous recessive, and homozygous recessive when it is small b, it will give brown color. When it is small b, it should ideally be brown color. Baat sahi hai na? So which means over here this one will be brown, and heterozygous definitely will be black because it has one dominant allele. So this one will be black, but this will be brown. So the question says that both will be black. No, one of them will be brown. So this is wrong. Assertion is wrong. The black color of the eye is dominant, absolutely correct. Here the question mate is given that the black is dominant. So over here black is dominant, which is correct. So assertion is wrong, but reason is correct. So we have to choose option D. Assertion is false, but reason is correct over here. Coming to the next question, the gene combination of purple flowered pea plant is denoted as capital W. This is the gene combination of pea plant which has purple flowers, and again purple is showing by capital W, which means it is dominant. So this is your purple, which is dominant. Now don't ask that, ma'am, why have they not used capital P, capital P? It is again the need of the question. Question might try to confuse you. Even though ideally if we were following Mendel's genetics, we would use capital P, but since the question tries to confuse you, they are using capital W, capital W. And that of the white flowered pea plant is used as small w, small w. So this is your white. Okay, when these two plants are crossed, F1 generation is obtained. Okay, definitely when you cross capital W purple in small w, it is basically going to give you the F1 generation, which will have capital W, small w, which means all will be purple in nature, all will be purple in colored flowers. Okay. List two observation made by Mendel in F1 generation. But Mendel in the F1 generation saw that all the offsprings, all the progeny were resembling only one parent. All of them were resembling only one parent because this one was purple, or this one was white. And he saw that when we receive the F1 generation, all of them are actually resembling only one parent. All of them are purple, so they resemble only one parent. That was the observation that he found, which means that the dominant trait is only able to express in the F1 generation. The dominant trait expresses in the F1 generation and the recessive one is somewhere hidden. That is what he observed in the F1 generation. Give the percentage of white flowered plants in F2 generation. Again, they are asking the percentage. So, you have to go back to calculating percentage. Got my point, beta? Let us try to solve this, no worries. Percentage of white flowered plants. Okay, in F2 generation. So, let us make a page. F2 generation may we have to find the percentage of white colored flowers. So, this is basically your F1 into F1 beta. Into F1. This is your both in purple. Both of them are actually purple flowers. And they are asking you the percentage of what? Of purple colored, white colored flowers. So, we have to simply first make the gametic combination. And let us see. Once we try to cross them, we will get capital W, small w. Again, they could also cross, so we will get capital W, small w. And finally, when they cross, they will form, finally, when these two will cross, they will form small w, small w. So, over here, beta, since all of them have one capital W, they will form purple flowers. And this particular one is actually responsible for forming a white colored flowers. So, the percentage of white flowers it is asking. So, we know that overall over here we see that there are 4 offsprings that we can see easily. So, out of which only one particular is white. So, out of 4 only one is white and you have to find the percentage. So, multiplied by 100, what you will get is 25%. You will get 25% to be actually the percentage of white colored flowers in the F2 generation, right? So, this is your F2 mein 25% is the answer. Ratio of gene combination of capital W, heterozygous, and again, I think this should not be capital W, small w, should be recessive, homozygous recessive. Let us see, what will be the genotypic combination? Genotypic ratio will actually supposed to be, you have to write for this one, homozygous dominant, heterozygous and homozygous recessive. So, we have only one that is homozygous dominant, we have 2 which are heterozygous. So, I am going to write 2 and then again we have one that is supposed to be homozygous recessive. So, the genotypic combination will obviously for a monohybrid cross be 1:2:1. Right, one difference between dominant and recessive trait. But a dominant is the one that will express regardless of recessive allele or not. But for recessive allele, in order to express itself, it needs to be in homozygous recessive condition. The same thing is written over here. All F1 will have purple flowers. The white flower will not appear because it is recessive. And in F1 only dominant expresses. Now, percentage of white flowers, again it will be 25% in F2 generation. Genotypic ratio will be 1:2:1. Same thing that I've just now explained it to you guys. Now, talking about dominant trait, expresses only one copy is present. That means it doesn't matter if there is recessive allele or not. It is going to express regardless of that. And recessive one is the one that will only express in case of homozygous recessive condition. That means there should be both the copies or 2 of the copies should be only in homozygous condition. It should be. Now, talking about dihybrid cross. With a dihybrid cross, mein di actually represents 2. So, it means that there is a cross that involves 2 characters that are being taken into consideration simultaneously. Let us try to understand this with example. So over here in dihybrid cross, let us talk about seed shape and seed color, and we are going to take both of them together. Let us see how. Firstly, beta, as we studied from the contrasting traits wala table, that seed shape could either be round or it could also be wrinkled. It could also be wrinkled. If it is round, that means it is dominant in nature, and if it is wrinkled, that means it is recessive in nature. Okay, and again, dominant has to be represented by capital alphabet, and wrinkled, which is recessive, has to be represented by small alphabet. So this is how you represent, okay, about the seed shape. Now talking about seed color could either be yellow or it could be green. Okay, these are the two different colors of the seeds. Now yellow, if it is there, it is actually dominant in nature, and you have to represent by capital alphabet, so you will write capital Y. And green, which is a recessive trait, you have to represent it with a small alphabet, so you will write small y. So this is how it is actually, taken into consideration about two different characters. Firstly, it is seed shape, secondly, it is seed color. Now talking about beta dihybrid cross, what did Mendel do? Firstly, he chose parents, and again, in case of parents, to make things easier for you, you should always go for homozygous pure parents. That means you have to take pure lines, okay? So I will consider one parent that is actually which has round seeds and yellow in color. This is the first parent, to make things easier for you. And the other parent is beta having wrinkled seeds which are green in color. Wrinkled seeds, and they are green in color over here. Now let us write the representation of how they will look like. Beta round could look like this also, and round could look like this also. Yellow could look like this also, and it could look like this also. But ma'am, out of these two genotypes, which one should we prefer then? Like we said, to make things easier, we will always go for homozygous pure. We have to go for homozygous pure. So we will go for this one because both of them, these two different characters, are in homozygous state. This is also homozygous dominant, and this is also homozygous dominant. So since we are just youngsters, we will go for easier things. So homozygous dominant is what we will prefer. Although yes, definitely this will also look round and this will also look yellow, but they are heterozygous, so we will avoid them to avoid any confusion. Got my point? So let us start now. For this one, we are going to write round is this. This is actually round, and this is yellow. Okay. Likewise, for wrinkled, you can simply write small r, and green ke liye you will write small y, small y. This is how it looks like for the parental combination. But now when they will form gametes, what will happen? This is where most of the students get confused. How will they form gametes? Let us try to simply think that when they form gametes, both of these R will separate. Both of these R will separate, and both of these Y will separate. Both of these Y will separate. But now there is a possibility that this R could go with this Y, this R could go with this Y, this could go or this could go together. But in any of the case, you will find that all of them will look like this, round and yellow gametes. Correct? Over here also you see there is a possibility, firstly all of them will separate. So these R will separate and this green alleles will separate. Again possibility mein kya hai? Any of these could go together. So again all of them will give small r, small y. So this is how you made the gametes first beta. Yahan pe we have made the gametes first very clearly. So I am simply going to write this capital R, capital Y because all the gametes will look like this, and over here all the gametes will look like small r, small y. Just let me write it a little above so that we have more space on the board. Capital R, capital Y will be the first gamete over here, and over here the gamete will be small r, small y. Now once they fertilize, because gametes are actually supposed to fertilize, that is what we know. But once they fertilize, what are they supposed to form? They will form a zygote which will have this condition, capital R, capital Y, small y. Baat sahi hai ki nahi? This is how they will look like. Some of you might think that, ma'am, okay, why don't they look like this? Capital R, capital Y, small y. We can also write like this. But we don't write like this. Reason is we have to put the same alleles together. So for R we will make a pair, and for yellow we will also make a pair. So we have to write it like this instead of writing like this. This is not a running alphabet wala notebook, so we have to mention that yes, there have to be pairing that takes place. So this is what we get in the first generation. Now beta, again, if you notice in the first generation, what are you going to find if we talk about the phenotype? How is it going to look like? What is the condition of this particular seed? It has one dominant, again, so it will look like round in shape, and it has again one capital Y, so it will look like yellow in color. So this seed is also again round and yellow. So in F1 over here, Mendel again saw that the F1 progenies, the filial one generation progenies that you see over here, represent only one parent. Represent only one parent. So Mendel was like, wow, what to do now? Let's go back to basics. So Mendel thought, okay, why not let's self them together. Why not we take two parents of F1 generation and cross them together? So that is what he did. He took two parents of F1 generation and crossed them together. Okay, so what will be the condition? We have to cross these two parents together now. This is the first parent, and then the second parent is also the same because it is F1. So now we have to cross them together. This is what he did in the second generation. But ma'am, now we have got this particular condition. How shall we form the gametes? How will we form gametes from such a typical genotype? Not to worry beta, I am over here to help you. See, the first genotype is capital, let us write again, F1 into F1 kar lete hain. So F1 into F1, over here we have both the parents. We are just making the gametes, okay, first. So we have in heterozygous condition for both the characters. This is what they actually look like. Simply do the thing that we just studied when we started doing this cross. When we made these gametes, we also did the same thing, the same pattern we have to repeat over here. Firstly, separate them. Separate capital R and small r. And then separate idhar se bhi capital R and small r. So both of them are now separated. Over here you separate capital Y and small y, and over here also you separate both of these alleles, capital Y and small y. Now see, there is a possibility that this capital R could fuse with this one. This could go together. So we will get the first gamete to be capital R, capital Y. This will also give capital R, small y. This will give small r, capital Y, and this will give small r, small y. And since both of them are same, so over here also you will get the same gametes. Let us still draw just to get any confusion clear. Okay. This is the gametic combination that you are supposed to get so as to proceed with the F2 generation. This is how the gametes will look like. These are only the gametes. Ab what will you do with the gametes, beta? We have to make a Punnett square. And now how will you make a Punnett square, ma'am? We have to make some boxes in the Punnett square. You see so many boxes, ma'am. How did we decide कि इतने सारे boxes we will form? Like I said, you simply have to count the number of gametes, beta. Over here you see how many gametes are there from the parent first. Parent first made how many gametes? 1, 2, 3, 4. So just write 4. From parent 1, these are the 4 gametes. Parent 2 form 1, 2, 3, 4, 4 gametes. So just multiply them and you will get the number of boxes. There will be 16 boxes in the Punnett square. I'm writing in PC, which means Punnett square. So the Punnett square will actually have 16 boxes with us. Okay. Again, so over here you see 16 boxes are present. One we will write that over here is the male gamete. Over here is the female gamete. Okay. Now let us mention the gametic combination. We had capital R, capital Y. We had capital R, small y. We had small r, capital Y. And we had small r, small y. These are the combination of the female, and the same combination of the male. Both of them are actually the same thing. So there is no confusion as such. Let us write again. Capital R, capital Y, capital R, small r, capital Y, and small r, small y. Now let us try to write all of them, beta. Let us see. When both of them will cross or fertilize, they will form capital R, capital Y, capital Y. Okay. Again, over here we see capital R, capital Y, small y. Capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. You don't have to simply see, you have to make it in a notebook also. So start doing it with me. Take a fresh page, even if you have not made notes, try doing it with me. Okay. Now again, capital R, capital Y, small y. Capital R, small y, small y. Capital R, small r, capital Y, small y. Capital R, small y, small y, small y. Okay, now capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Then again we have capital R, small, no, we don't have capital R. We have small r, capital Y, capital Y. We have small r, capital Y, small y. Okay, again we will do over here capital R, small y, capital Y, small y. Now we have capital R, small y, small y. Next up we have small r, capital Y, small y, and finally we have small r, small y, small y. These are the different type of genotypes that you are supposed to see. Over here you got 16 genotypes, what we have written over here. Got my point? Now beta, let us try to see what each genotype will give as a phenotype. Okay, this has both dominant, so this will be round and yellow. Round, yellow. Okay, again we will be over here having round because it has both the capital R, and again one is there which is capital Y, so round and yellow. Over here again we have round and yellow. Okay, then again we have round and yellow because we have one dominant allele each over here for both the characters. Okay, then again over here we will also have round yellow because round is in homozygous dominant and yellow is in heterozygous. Over here we will have round but green, so let me write round but green because this particular over here is in recessive condition. Next up we again have round and yellow. Round yellow. Again we will have over here round and green. Okay, because this is over here in homozygous recessive condition. Next again we have round yellow because both of the characters over here have one dominant allele each. Round yellow yet again because again they have dominant alleles. Next we have wrinkled but yellow beta. We have wrinkled but yellow. Let me choose another color. Over here we have wrinkled because small r, so it will give wrinkled, and then capital Y will give yellow. Then over here again we will have wrinkled but yellow. So we have wrinkled yellow condition over here because this will go for wrinkled and one capital Y will give yellow color. Now this will again look like round and yellow because both of them still have one dominant allele each over here. Then this will be round and green because capital R will give round and both of them are small y, so they will represent green. Okay, then we have wrinkled but yellow. We will have wrinkled but yellow because this is supposed to give wrinkled, small r, homozygous recessive, and yellow because there is one capital Y. The last condition that you are going to see it will give you beta, it is supposed to give you wrinkled but green. It is going to give you wrinkled and green. So we are going to write wrinkled and green. Once in dihybrid cross, F1 generation was resembling to the dominant parent. But in case of F2 generation, there are new characters that are also seen. Apart from both the parents, there are new combinations also seen. So round and yellow was already present. Round and yellow, which was dominant beta, it was already present. We have to actually write the round and yellow number. Let us see round and yellow kitne hain. Count it. Round yellow 1, 2, 3, 4, 5, 6, 7, 8 and 9. So we have actually 9 round and yellow combinations over here. So we have to simply mention that 9 round and yellow we have, and this was there in the parental combination. Parent was also round and yellow. Now let us look at round and green. Round and green, how many are there? Let us try to calculate. Count round green and round green. So basically there are 3 that are round green. Okay, there are 3 that are round green in nature. Now talking about wrinkled and yellow. How many are wrinkled and yellow? If you see closely over here, you will find 1 wrinkled yellow, 2nd wrinkled yellow and 3rd wrinkled yellow. So there are ideally 3 wrinkled yellow that are easily visible. Talking about wrinkled green, how many wrinkled green are present over here beta? Only 1 wrinkled green is over here that is being seen. So that means that there is 1 wrinkled green which is also parental because parent was also wrinkled green. So these are the parental combinations. So in F2 generation, you saw that there are round yellow which are parental and wrinkled green which is parental. But over here, these two are absolutely new. They were not seen in the parent. So that is the difference, that in F2 generation, new combination of characteristics are also seen. New combination of characteristics are also seen, and this is actually also representing your dihybrid cross में phenotypic ratio of the F2 generation. This is the phenotypic ratio of dihybrid cross in F2 generation. Okay, 9:3:3:1 asked in exams many times. But the more important thing is that there are new combinations that are being seen. For example, beta, you also have some characters which resemble your father and which resemble your mother. You can have your eyes from the mother, you can have your nose from the father. It is not that your body is complete replica of your mother or your father. It has characters from both of them. There are new characters. For example, you might get black colored eyes from your mother, but you can also get a very tall height from your father. So you have new combinations, right? You have a different combination. Likewise, over here in F2 generation, we find different combination of plants that are also seen, and that is explained very well with the help of law of independent assortment. Law of independent assortment, the names, even though it look very overwhelming, what is independent, what is assortment? Independent means separate, something that is not connected, and assortment means again to segregate. Think of it, for example, if you are in grade 10th, you are friends with your close person, okay, whoever is your best friend, you are very good friends with them. In class 11th, if you go and choose science and if your friend chooses commerce, so both of you will go in different sections, in different classes in 11th and 12th, in different colleges, in different careers, different professions, right? That means even though you were at some point together, but now you are segregated, you are working independently, but you are still friends. Likewise, in case of law of independent assortment, if we have two genes, both of them will move independently. Both of them will move independently. They are not stick to each other. Likewise, this is why you are able to see that you have certain characters from mother and father. Because if their genes were sticking together, how will you get eyes from the mother and height from the father? So they are segregated, they are separated, they are independently moving to the next generation, and no two genes are linked. The inheritance of one gene does not affect the inheritance of any other gene. They are moving to the next generation independent of each other. That is what is the law of independent assortment. Next, coming to the topic sex determination. Last topic and very easy, beta. What do you understand by sex determination? It simply means to know whether the child, whether the offspring or the progeny will be a male or a female. What will be the sex of the child? It is called as sex determination. Now, there are different ways how sex is being determined in different organisms. In some organisms, the temperature, their environmental condition decide the sex of the particular organism. If the temperature changes, the sex of the organism also changes. Yes, it might be very surprising, but it is true. So, external factors decide what is going to be the sex of the organism. But that is not in case of the humans. In humans, the sex is actually determined by the gametic combination. The gametes received from the egg and the sperm and the zygote formed. And this is not decided by environment. This is not changing by the environment or the temperature. It is not that if today temperature is 50 degrees, so you are a male, and if temperature drops to 4 degrees, you are a female. That doesn't happen, right? So, it is very genetic in case of humans. Talking about humans, firstly, beta, they have 46 chromosomes, as you know by now. Humans have 46 chromosomes, or we can also say that there are 23 pairs, one and the same thing, na? You say 46 chromosomes, or you can also say 23 pairs. It is one and the same thing. Now, beta, out of those 23 pairs, 22 pairs are autosomes, which are common in males and females. And one pair is actually called as the sex chromosome. One pair is actually called as the sex chromosome, that is different in males and females, and it also decides the sex of the person. I will repeat once again that in these 23 pairs, 22 pairs are called as autosomes, which are the same in males and females. Same in males and females, there is no difference, okay. These are the same, they are called as autosomes. Got my point? Okay. Now, the other one remaining left pair is called as the sex chromosome, and this sex chromosome is actually determining the sex of the person. So, it is different in males and females. In males, it is XY, and in females, it is XX. It is XX. Got my point? So over here sex chromosome is different because it determines the sex of the person. That is why it is called as sex chromosome, or it is also called as allosomes. Let us try to understand this better with the help of an image. See, there over here you can see that there are 23 pairs of chromosomes. There are overall 23 pairs of chromosomes, out of which 22 pairs are beta autosomes. That is all those that you can see over here in blue color, they represent your autosome, and there are 22 pairs of autosomes which are going to be same in males and females. Got my point? The other one left out pair, that is called as the sex chromosome. Now sex chromosome actually varies. If it is XY, it is the male, and if it is XX, it is the female. So let us say the person is a male. So male beta will have all these 22 pairs, including this XY. Including this XY, this will be the composition of a male. Correct? If it is a female, so it is supposed to obviously have all these 22 pairs of chromosomes, including this particular XX. So this will be the condition of a female, which will have XX in her genetic combination. This is how the sex chromosomes are being differentiated in males as well as in females. Now talking about how sex determination actually takes place, for which again we have to consider the parents first. What will be the combination of the parents? In case of parents, the sexual combination of the sex chromosome is actually XX and XY. Okay, one is supposed to be XX and XY. XX is actually the female and XY is actually the male. So again we are going to write, this is a female, and over here we have a male. Now the next step would be to form the gametes. So yes, definitely they are going to form the gametes over here because they are required in sexual reproduction. So how will the gametes look like? In case of female, the gametes will be containing only and only the X chromosome. Let me use a different color over here. The gametes in female will only contain the X chromosome. This one will also have X and this one will also have X, because females only produce X chromosomes. So they can produce only X chromosome in the gametes. Whereas in case of males, there will be one gamete that will have X, I mean half will have X and half gametes will have Y chromosome. Now beta, what will happen when fertilization takes place between the egg and the sperm? There could be a possibility that this X could fuse with this X, so it will give XX. Again, this could fuse with this also, it will give XY. This could fuse together, giving XX. And if these two fuse together, they will give XY. Now over here, let us write the phenotype and the genotype. If it is beta XX, it is supposed to be a female. Again, over here it is XX, so it is again supposed to be a female. And talking about XY, it is supposed to be a male. So this will be a male and this will be a male. So over here you see, whenever sexual reproduction takes place, there is a chance that whenever a child is born, 50% chances take place of being a female and 50% of chances are of being a male. So which is why we never can predict, and we can also never blame the mother entirely for producing a female child, because it is not in the hands of the mother. Mother is only producing the X chromosome. Female is only producing the X chromosome. Now depending upon what does the sperm carry, whether it is an X, whether it is a Y, it will determine the sex of the child, right? But again, in case of any sexual reproduction, I will repeat that 50% chances are of being a male child or 50% chances are being of a female child or a girl child. So simply we have to write the phenotypic and the genotypic ratio. Let us write the phenotypic ratio once. What is the phenotypic ratio? See, phenotypic ratio, it will be between males and females, or boys and girls you can also write. Males is to females, what can we write? How many beta males are there over here? We have one male, we have two males. So 2 we can write. Females also 2. So we can cancel, we get 1:1. So phenotypic ratio is supposed to be 1:1 in this particular case, okay? Talking about genotypic ratio. Again, genotypic ratios will be very similar to this one only, because we simply have to write the genetic combination. Genotypic can be either XX or it could be XY in case of males. How many XX are there beta? 2 are there, and XY again 2 are there. Cancel them out, we get 1:1. So the genotypic combination over here will be again 1:1. So the phenotypic ratio or genotypic ratio you can see over here, both are same, both are 1:1. That means again the chances are 50% of a boy child or a female child. That is how it is also represented in the form of a Punnett square, you can see easily. Now the question whether comes in the form of proving it, you simply can draw a Punnett square to represent things very nicely. Talking about some questions that we can solve now. Question says a tall pea plant with round seeds, they have given you the genotype also to make things easier for you, is crossed with a short pea plant with wrinkled seeds, the F1 generation will be. I think by now you should simply be able to solve it verbally also, but let us still draw for easier understanding. When a tall plant with round seeds is crossed with a dwarf plant with wrinkled seed, what will be the F1 generation? Gametes over here will be capital T, capital R, small t, small r. When they will form the F1 generation, they will form capital T, small t, capital R, small r. So all of them will be tall, heighted and having round seeds. So again they are resembling only one parent. So 100% are tall and round. 100% are tall and round. So we can see 100% tall with round seeds. Option D is the correct option over here. But please mention if in case they don't give you the genotype in the question, you have to figure it out. You have to make it out on your own. But they will definitely mention that whether the plant is homozygous, a pure line or something like that, so that you can deduce the genotypes as well. Now we have a case-based question, pretty long but very easy, not to worry. In human beings, there are 23 pairs of chromosomes. We just now studied, very good. It is 23 pairs or 46 in number. Out of these 23 pairs of chromosomes, that is 46, 22 are basically called as the autosomes, that is same in both males and females. And a pair of chromosomes are actually called the sex chromosomes. The sex chromosomes are of two types, X and Y. The sex of a child is decided at the time of fertilization or the fusion of the gametes. In other words, at the time of zygote formation, chromosomes get inherited from the parents of a child. Okay. What are chromosomes? After all of this, they are asking you what is a chromosome. So what are chromosomes, beta? They are actually thread-like structures that are present in the nucleus of the cell. We just started in the beginning of the lecture also that they are, extremely elongated structures. Depending on the stage of the cell, it could be either packed tightly in the form of chromosome, it could be loosely present as chromatin. They have DNA as well as proteins, and DNA has genes, which are the units of inheritance. Why is the pair of sex chromosome in human male mismatched? Which means to say Y is in males one X and one Y. Why not like females XX, or let us say YY? Why not that? The reason is, beta, there are certain genes that are present on the X chromosomes in males are not present on the Y chromosomes and vice versa. So we need two individual chromosomes in males. If all the genes were same, it would be either XX or it would be either YY. But certain genes are supposed to be on X, so it has X chromosome. Certain are supposed to be on Y, so it will be Y chromosome. They are different. Show with the help of a flow chart that the statistical probability of getting a boy or a girl child is 50-50. So again, you have to draw these diagrams, beta. You can either draw this and mention the phenotypic and genotypic ratio and write it down that the ratios prove that the chances of a boy child or a girl child are equal. And likewise, you can represent with the help of a Punnett square also, which is mentioned over here. So either of these two you can use to write down your answer and describe it very precisely to get good marks. Same thing is written over here. ये मैंने यहाँ पे लिख देती हूँ. You can refer to the previous slide. Previous cross. Okay. That we just draw. Okay. Now the same thing is written that I explained. So you can take a screenshot, or I think you will get the note. Not I think you will definitely get the notes. So please make sure to refer to it so that you can solve certain questions like that. Question number 10 says, a pure pea plant having round and yellow seeds. Round and yellow seeds, and the thing, pure pea plant but not genotype. But pure means homozygous. So pure which has round and yellow. Round and yellow. Okay. The pure one cannot be this, because this is not pure. This is hybrid. This is heterozygous. So this is not pure at all. Okay. Is crossed with another pure plant having wrinkled and green. So wrinkled and green will look somewhat like this. This is wrinkled and green. Subsequently, F1 progeny is self-pollinated to obtain F2 progeny. Cool. But they are asking the seeds of F1. So again for F1, make the gametes capital R, capital Y, small y. So when they cross, capital R, capital Y, small y, which means all will have round and yellow seeds. In F1, this is your F1 condition. So F1 will have all the round and yellow seeds. Got my point? Give the possible combination of traits in seeds of F2 generation, also give the ratio. All the possible combination of traits in seeds of F2 generation and the ratio, beta, it is simply asking you this particular thing. They are simply asking you this. So you have to draw this and show that what are the different combinations. How does it look like? And then please also don't forget to mention the phenotypic ratio 9:3:3:1, which they are asking in the question. Okay, so that is the correct answer for this particular point. Then we have, okay, state the reason of obtaining seeds of new combination of traits in F2 generation. It is that laws, basically it represents your law of independent assortment, that genes are not tied to each other. They can, move independently and they are not inherited together. So any inheritance of one gene will not affect the inheritance of the other gene. So this is the same thing. All the F1 will actually look round and yellow because both have dominant traits in them, both are in heterozygous. B, we have just seen the dihybrid cross. New combination occurs because of the independent assortment of genes, that is during gamete formation the gene will separate independently of each other, they are not linked together. Now we have the last question of the day and it is supposed to be a homework question, which is I think a ritual of the class now that we always do a homework towards the end of the session. Okay, so please let me know the answer of this particular question in the comment box and the chat box to just let you know that if you have understood the concepts properly or not. Okay. I think we have so far covered all the topics of the chapter, but if you still have any questions or doubt, you can drop it down in the comment section. And if you feel that certain concepts are unclear, please go back, rewind and watch it once again to know and strengthen it up. That is all for the session today. I will see you in the next one. Thank you so much for watching, students. Please take care of yourself. Keep studying. Keep working hard. Make your parents proud. Stay hydrated, eat well, and I will see you in the next one. Thank you so much for watching.